This week in Evolution, episode number 2, recorded on October 27th, 2015. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiEVO, the podcast about the biology behind what makes us tick. Joining me from Salt Lake City, Utah, Nels, Elde.
Hi, Vincent. Great to be here again.
Got your name right.
Yeah, we're making progress. We're evolving.
Yes, evolution is important. I'm very sorry I got it wrong for most of last. I did a podcast at Ann Arbor on Thursday at TWiM, and I got one guy's name wrong the whole episode.
Well, no sweat here. I've been called much worse.
Me too. You know, names are hard.
That's right. Yep.
So it's been a month since we last spoke. Are you progressing? Are you getting results? Are you publishing papers?
Yeah, I'd say that's a tall order. I'd say a little progress here and there. I was actually in your neighborhood not too long ago. I gave a seminar to the microbiology department at Yale, and so I was in Connecticut for a few days. Looks like you've been doing some globetrotting as well.
Yes, where have I been? Latest trip was Ann Arbor, University of Michigan. I did a TWiV and a TWiM there. I was in La Jolla, did two TWiMs there. That was very interesting. And then I was in Germany. I don't know, when we last talked, had I gone to Germany yet? I don't remember.
I think this was before you had left. So, active month.
I spent 5 days there.
Cool.
And I'm going to be on the West Coast in about a week and a half. I'm going to Portland. I was invited by students to give a talk at their retreat.
Oh, that should be fun.
And there's just a few minor trips. I think that's it for 2015. Not a big traveling year. I didn't hit 50,000 miles.
That's the cutoff for
gold.
Gold Club, right?
I did it last year, but I had gone to Brazil and Australia. That'll do it,
There you go.
I'm going to end up with like 42,000. It'll be silver. It's fine, though. I still get to get on second. I don't care when I get on, as long as I get free baggage, because I have to check the TWiV studio.
Oh, yeah. There you go.
You traveling besides Yale? Anything else?
Quite a bit, yeah. So I'm going to hit Colorado, kind of a local visit in a couple weeks with a friend of TWiV's, Sarah Sawyer, is having me out. So she just moved her lab from Austin to Boulder. Catch up with her and see how things are going. And then I'm going to hit a little farther up the West Coast, Seattle, in December for a talk to the microbiology department at University of Washington. Mm-hmm. And then we'll conclude 2015 in San Diego with the trip to the ASCB, American Society for Cell Biology annual meeting.
Wow, busy guy.
Yeah, we're past the gold on this year.
So we're going to get, besides this, we'll get 2 more TWiEVOs in for 2015, right? So we'll have 4 in all. Yeah,
that'll be a good set for getting up and running, and then we'll go into the new year strong.
Great. Now, you sound really good this time, Nels. What happened?
Yeah, well, I guess this is the evolution of a podcast, so to speak. So I'm trying to follow your lead, just mimic your style here. So I got a new microphone on your recommendation in Elde Lab Studios. We're not quite as sophisticated as Racaniello Lab Studios, but we're doing what we can. We also, I think we're coming along nicely, both with theme music and a logo. So this thing might start to look official pretty soon.
Yeah, that's great. Yeah, you have to do all these little things, and then we'll put this on iTunes, and we'll build a website, which could happen soon, certainly before the end of the year. But I'm targeting sometime early in November to launch this baby.
Yeah, that'll be fun.
Yeah, my studio is a little more complicated, but this is kind of the nerve hub of the Twix empire. So
yeah, growing empire.
Got lots of Skype connections, and I'm always thinking about how to reroute the wires. I have so many wires on my desk. I need a bigger desk now. There's no room to write, and I need to write. Actually, I don't have to. I just need to type, right?
There you go.
Speaking of typing, are you into keyboards?
I certainly use a keyboard as a grant writer, which is, I would say, a good portion of my job, maybe more than 50% of my job in some stretches. Yeah.
You probably use the keyboard that comes with your machine, right?
I do. Yeah, just on my laptop.
Yeah, I like keyboard. I like clackety keyboards, really clicky clacky ones with switches.
Oh yeah,
but
the old style. Yeah.
I can't use them to podcast because it has to be quiet, and I'm often searching for things. So I have a crummy keyboard that's silent, but I lust for a clickety clack one.
You can always dream big, I guess.
So the music that will be on this podcast, you just picked. It's by a band you picked called Trampled by Turtles, right?
Yeah, exactly. So this is a bluegrass band, or I guess they promote themselves as an alternative band that I've been following for some years. It's a Minnesota band. These guys are from Duluth, Minnesota originally. And I'm guessing they're now platinum or diamond flyers with the world tours that they go on promoting their music. I've seen them around recently. They were in Salt Lake this summer, and they hit the Telluride Bluegrass Festival from time to time. Just really good sound, I think, and energy. I thought it would be a fun way to kick off and conclude our podcast with some bluegrass instrumental.
So this is Minnesota where you grew up, right?
Yeah, exactly. Okay. So I grew up in Minnesota, and then as we talked about yesterday, sort of have been migrating around Chicago, Seattle, and then currently in Salt Lake City.
And you also made a logo, which is cool, and people will like it a lot, I think.
Oh, thanks. Yeah. So this actually, gets to some of these ideas we were talking about again last week in the origins of sort of evolutionary thinking. And so we took a butterfly that is one that you could find in the rainforest, something that Henry Walter Bates might have seen when he was first thinking about mimicry back in the 1850s. And then juxtapose that with a virus crystal structure, which if you put it back to back, I think resembles a little bit of a butterfly. It's trying to hit the idea of evolution kind of happening at all of these different levels of biological organization. The idea of how copying things can actually be a pretty good evolutionary strategy in many contexts, and kind of also catches the eye, I think.
Nice, pretty cool.
Yeah, thanks.
It's very cool. And speaking of evolution, well, that's what we do on this podcast, right? My last TWiEVO out in Ann Arbor, one of my guests was Adam Loring, who you must know. Right?
Oh, yeah, of course. Adam's a great guy.
Who is a recently minted assistant professor, probably a little more recently than you, right? You're a little more experienced.
Yeah, that's right. I think, has Adam had his lab maybe 2 years now?
Something like that. I think he's had a paper on error catastrophe of influenza virus.
Yeah, some really interesting work.
Pushing it over the edge with an antiviral. So we talked about quasi-species and error thresholds. We didn't have a lot of time. We had about 15, 20 minutes for each of three guests, but
oh yeah,
that's cool stuff. He doesn't like to use the term quasi-species. Did we mention that last time?
I can't remember if we did, but
we might mention it today.
I think it will come up, and actually the topics you're hinting at in Adam's work will, I think, show up today pretty nicely in the paper that we picked to cover.
Hey, I like this idea of massively sequencing to find out what variation really is. And I don't know if you've seen this. A paper just came out, I think, in Cell, where they looked at VSV variation in the yield of single cells.
Oh, I haven't seen that yet. Thanks for the tip. I'll take a look.
Take a look at that. I'll probably do it at some point on TWiEVO. But cool stuff. Anyway, let's get into today's paper. What do you have for us?
Yeah, so, this paper, the idea for this paper, which is on the measuring or trying to measure mutation rates of HIV virus, kind of has two motivations. The first one was I was actually just talking to some friends. So as part of the travels in the last month, I stopped in Minnesota and was discussing our new podcast with some of my best friends. And they said, "This Week in Evolution," and they started laughing because they, when we think about evolution, we often think about the order of millions of years or hundreds of millions of years. And how could something happen in a month?
Yeah. It's true. But, viruses shattered all that, right?
There you go.
I mean, we used to think about dinosaurs in millions of years, but then viruses do it in like 5 seconds.
Exactly. Evolution can move faster than we think, or commonly think. And so that's one of the motivations for this paper, is to really, even if you sample in one day people who are infected by HIV, we see an incredible amount of diversity, as we'll discuss in a bit of detail here. And so I think just as a general theme, it's really good to think about evolution not as sort of a historical, this happened hundreds of millions of years ago, just because, we don't see dolphins sprouting new legs or anything like that. If we just look a little bit closer, especially at the microbial world, wow, there is all kinds of variation popping up all around us and constantly. And so evolution is really an active field, both of study, but also of just as it unfolds really right before our eyes.
I like to tell my virology class, I like to give them these little crystallizations of viruses. And one of the things I say, I have on a slide, it's a picture of Earth. It says, at this moment in time, there are 10 to the 16th HIV genomes on the planet.
Wow.
And what does that number mean? It means that there's already resistance to all the antivirals we have and anything we would ever develop. That's how big that number is.
Yeah, it's really amazing. Mind-boggling, really.
It's just incredible. And that's just HIV.
Exactly. Just a tip of the iceberg. So, and then the second motivation, Vincent, for today's paper is just to think about mutations in terms of evolution. This is sort of the, nuts and bolts, the bread and butter of the evolutionary process is mutation. And so, if we just look at how the entire field is defined, population genetics, for instance, which is really synonymous with evolution, one simple definition is that this is the study of the distribution and change in frequency of mutations within populations. And so just by understanding how mutations accumulate, how their frequencies change in populations, some really clever scientists over the last 100 years have learned some really powerful things about how evolution unfolds and principles underlying that. I was going to quote just the first sentence of a different paper I found, which is measuring the mutation rates of not HIV virus, but actually of yeast. But the opening sentence, I think, is really kind of crystallizes it. It says, mutation is the ultimate source of all genetic differences within and among species. And so that source of differences, that's what is responsible for all of the variation around us. And so this is sort of a core principle.
Nels, just occurred to me, we use this word mutation, right? So a mutation is a change from some consensus, right? But who's to say what the consensus is, right?
Yeah, that's right. Great point. And who's to even say what the mutation is? So today we will be talking about point mutations, that is changes in DNA base pairs relative to, as you're pointing out, a consensus virus sequence in this case. But mutations could take on all kinds of flavors. That could be a gene duplication or a massive loss to part of a genome, big time rearrangements. And so again, you're right. So what these are relative to and even how they unfold are very different things from time to time. And so it's this massive source of diversity.
In humans, we actually often we use the word single nucleotide polymorphism, right?
Correct.
Which I think has implied that it's not necessarily a mutation because you don't know which one is good and which one is bad. It's just differences, right?
Exactly. And that gets at another point, I think, about the definition of mutation, which is it's good to leave in popular language, mutation takes on a negative connotation.
Yeah.
But it's kind of good to leave, if you can, to leave that sort of intuition behind and be more agnostic about what a mutation might be. It could be deleterious, as we kind of think. It could be neutral, or in rare cases, also beneficial.
Can we call them just variants?
Yeah, so that's, the language, people do that. The language, and variant is, I find myself saying that a lot. The polymorphism that you brought up, the SNP, or single nucleotide polymorphism, that's another sort of more neutral term that reflects, what really is, quote unquote, wild type or healthy. Right. In many cases, we're still just trying to figure that out, and it can depend on the context. Yeah.
Well, a human, basically every cell in a human has a slightly different sequence, right?
Absolutely.
So who's to say what wild type is?
Yeah, that's right. Exactly.
This is the concept of species. Originally, I think the idea was a species was something that had a single sequence, and then with virus variation discovered, proposed by Manfred Eigen and shown experimentally by Esteban Domingo and others, That's why they came up with this term quasi-species, because it was clearly not a single sequence. But now we know that animals aren't even single sequences.
That's right. We could call ourselves quasi-species, sort of on another level as well. Absolutely. Yeah. Yeah, and so I think that kind of captures sort of the challenge that actually we'll face to some degree today, is that what are we measuring everything against if that's sort of a complicated and moving target? And so I think as scientists, and as we'll see, as evolutionary biologists and microbiologists, virologists, that you sort of do the best you can with the yardsticks, but then everything is sort of relative. And so
one last comment before you dive into this. So basically changes, variations, or single nucleotide or multiple nucleotide changes, variation, let's say, is the basis for evolution. It can't happen without it. And we use viruses because it can happen very quickly, as opposed to humans where a generation time is 8 months.
That's right.
and you can't do experiments anyway. But, you can go to a mouse that has a faster mutation time, generation time, but there's nothing like viruses, right?
You're right. They really scream along. Nothing mutationally moves faster. And as we'll see, it might even be, from today's paper, it might even be faster than we anticipated in some scenarios. People, if we look at the history, of the study of mutations, some people will hearken back to the 1920s, and actually right in your neighborhood in Columbia University in Thomas Hunt Morgan's fruit fly lab, where they were doing experiments looking for spontaneous mutation in fruit flies. And even fruit flies, which are, relatively short generation times, and you can generate fairly large populations of flies quickly because they're so small, there were real problems with identifying mutants or genetic variation because the spontaneous rates of mutation were so slow. Folks like Calvin Bridges was a master of finding these, what he called sports. These were flies that looked a little different. Their eyes were different. Their wings were different. And you could pick them out.
Yeah. And then once you had them, that he would give them away and other people would study them, right?
Yeah, exactly.
And I remember in college we had a Drosophila lab. It was taught, by the way, by Jerry Fink. He used to be at Cornell. He's now at MIT. And I love those flies with the red eyes. It was so cool.
Yeah,
right?
beautiful.
Do you ever see a red-eyed Drosophila?
Oh, yeah, of course.
Just gorgeous.
Yeah, stunning. So that, one of the big breakthroughs was when this fellow Herman Mueller came along, and he was blasting these fruit flies with X-rays. And all of a sudden, these sports or these variants or mutants started just popping up all over the place. And what he was doing was introducing a source of new mutations. And now this was a big breakthrough, actually, and this is before we knew the molecular basis of what genes were, that DNA was the genetic material that doesn't come along for another 30 years. And yet now with this, by inducing these mutations, these geneticists started making some real breakthroughs and being able to... Where did
he get the idea to use X-rays?
That's a great question. I don't know offhand.
It's one of those paradigm-shifting things, right?
That's right. I do know that Thomas Hunt Morgan he was doing this, and then Morgan got onto it and recruited him to Columbia in order, because they could kind of see how this might be powerful if they applied it to the great fruit fly system. And so we teach this to our incoming grad students, or remind them of some of that history as part of the genetics class. And the reason is because it gets to the sort of fundamental basis of mutations being kind of the workhorse of variation. And this also ties into evolution. And so as geneticists, as Mueller and Morgan and these other folks started to cause mutations with using mutagenic agents like X-rays, they're able to really accelerate their search of what genetic variation can do. I would say as evolutionary biologists, we look back and try to take advantage of all of the variation that's already out there and has sort of been springing up over, more than 1 billion years. And kind of in parallel, we try to understand sort of the basis of that variation, what's important, what's not important, how it moves through populations, how the frequencies of variation change. And so, there's this kind of interesting parallel line of geneticists kind of mimicking the evolutionary process by inducing mutation, and then evolution having this sort of long history leading up to today and beyond, where these changes are just unfolding right before our eyes.
So in nature, we don't well, we do have X-rays, but we have other mutagens that are out there, right? But what's the main creator of mutation?
Yeah, so, there are both internal and external forces at play here, and this, again, will kind of factor into the paper, as we'll see. But, UV rays are a big source. And many what are considered carcinogenic agents, where you might get more mutation than you bargained for and end up with things that can cause trouble.
Yeah, my pick is related to that today.
Yeah, I saw that. I think I was trying to preview that a little bit. We'll get back to that in a few minutes.
I did that on purpose.
I love it. Yeah, it's great. So why don't we dive into the paper, Vincent?
Okay, I'm sorry I asked a lot of questions.
Not at all. This is fun. This is why we're doing this.
You're the expert, so I might as well take advantage of it.
We'll see if we can stump the expert. I don't think it will take very long. So the paper is called "Extremely High Mutation Rate of HIV-1 in Vivo." It was published, I think, just a few months ago. Came out in September, mid-September. Published in PLoS Biology, Public Library of Science Biology, and the group is headed by this fellow Rafael San Juan in Valencia, Spain. The San Juan lab has worked with another great virus evolution lab, Santi Elena's lab. I think you might have trained with Elena, and then has had his group in Valencia for a few years, and they're just churning out really cool work, especially thinking about things like this, mutation rates of viruses. And so I'd say we should dive right in.
Yeah, let's do it.
Yeah. So the first point to bring up is just why would we look at HIV virus? And in fact, it has been gained a lot of attention for obvious medical reasons over the years. And so there are all of these resources and possibilities to study it that I think make it an attractive source. And also because it's such a prevalent pathogen representative of this huge group of retroviruses, it's really important to understand how it mutates. I'm curious, Vincent, have you ever studied HIV? I know you've mostly focused on poliovirus for your career. Were you ever tempted to study this virus that we're talking about today?
I have not. I've been surrounded by people who have. Right here at Columbia, Steve Goff, of course, and Scott Hammer and others, but I have never. And, it's interesting, when I came here in 1982, a year later, AIDS was discovered as a clinical entity, and there was a lot of buzz, right? But, it's in virology, you have your virus, and I hadn't worked on retroviruses, so yeah, it kind of keeps you from going into other viruses because you're afraid that your study section, for example, will say, well, this person wasn't trained in retrovirology, so we're not going to fund this grant. So that really does it keeps people from moving around. However, you're lucky because you're an evolutionary biologist. You can work on any virus you want.
Yeah, so that is, I'd say it's one of the fun things. It's also, again, a challenge because if you're not careful, you can get spread too thin. So I kind of made a pact with myself early in my postdoc that I was going to avoid HIV evolution just because a lot of great people are doing it.
Yeah.
And of course, you always end up eating your words in these cases. So my lab currently does have an HIV project, partially just because of the neighborhood we're in. So there's a great biochemist and virologist, Wes Sundquist, here at the University of Utah.
That's right.
And so he pulled us in on a really fun project, and we may get to that down the road here on TWiEVO at some point. But why don't we circle back for today's paper, where they're really thinking about what's the mutation rate of HIV. And in fact, you might think, well, that question's probably already been answered given all of the sort of attention that HIV has garnered recently. But in fact, not everything is known, as the authors point out. And they try to make a case that, in fact, in some ways we may have been underestimating the mutation rate of HIV, just given the way that we're measuring how mutations accumulate.
How would you measure mutation rates? This has been done for many viruses and largely in cell culture or in vitro, right? So just how could you do that?
Yeah, exactly. So you're absolutely right. Many viruses have been measured, and it's kind of, I would say it's a patchwork of different methods over the years. And one reason viruses have been focused on a lot is because they have such small genomes. And so actually you can sequence without too much trouble the entire genome. And so the idea would be to take, depending on the type of virus, to take and sample a population. For a virus like HIV that has such a high mutation rate, you don't have to wait long. And as you were mentioning before, this idea of quasi-species, you can already, if you just sort of dip into the bucket and pull out virus sequences, you'll already see all kinds of variation. There's not a single genome that matches another genome in many of these populations because the mutation rate is so high.
So basically, I remember when a long time ago in my career, Peter PLoS published a paper where he picked 100 plaques of polio and 100 plaques of flu and sequenced one gene of both, right? By the old kind of sequencing, which was consensus sequencing, where you're really looking at the average sequence of the population.
Yeah.
And found no mutations in the polio genome in one of the capsid coding regions and a bunch for influenza virus. But it didn't allow him to quantify mutation rates. Other people have used polymerase assays, or nowadays you can do deep sequencing to do this. But let's say if you looked for a phenotype, Nels, would that be a bad way to quantify mutation rate?
Yeah, so if you're interested in sort of the evolutionary population genetics view of mutation rate, then selecting on a phenotype or picking a phenotype is actually a potential trap. And the reason for that is because the idea is that you would just like to know the frequency by which these mutations are accumulating in a population without the influence of outside variables like selection. And so
a big one.
A big one. There you go. And so if you're looking for a phenotype like a bright red eye of a fruit fly or something about the virus being able to replicate, then you're potentially biasing basically what the mutation rate really is, because you're looking at sort of a subset of things that are being inherited or selected for. And so that's kind of one of the challenges to mutation rate estimation, is to sort of pull that aside. And as we'll see for this new paper on HIV evolution, they did a couple of tricks to try to get around how selection might be acting and to try to get this new estimation in vivo from sampling HIV from patients.
So they have in their abstract first of all, right now the best way to look at this is by deep sequencing, right?
Yeah, and that's a good point. So moving from the PLoS days of picking one or two genes and sequencing that gene, given how hard it was just to get. I mean, don't get me wrong, that was the state of the art when he was doing that work and others were doing that work. But you get sort of this narrow window of how mutations are accumulating. And this has been another one of these, massive breakthroughs of the genomics era, by being able to deep sequence and get entire sequences not only of viruses with relatively modest-sized genomes, but also all the way up to humans. We now have this new view of being able to count mutations. And so for humans recently, there's been some new estimates of mutation rate in our species based on taking two parents and their offspring, these trios, and sequencing all three genomes, and then asking how many spontaneous mutations are there between the parents and the child that are not inherited from the parent but have arisen de novo in the child. And because you have all three data points, you can actually now triangulate and make that calculation in new and powerful ways.
What's the number they come up with?
I think you stumped me. It's not that many. I think on the order of something like 100 spontaneous mutations per generation, something like that.
That's not much, because the genome is 3 billion base pairs, right?
That's right. And so this is this is sort of a point mutations, and it's maybe taking a narrow definition of mutation. So a lot of potential structural variation and regions that are not easy to sequence. So when we talk about sequencing a human genome, we're not covering all 3 billion bases. We're getting closer and closer, but there are a lot of regions that are still kind of dark matter and are very hard to map and sequence. But it's a surprisingly small number for us, at least in current estimates.
In the virus world, we make a distinction between viruses with RNA genomes and viruses with DNA genomes. And in general, with a few exceptions, the viruses with RNA genomes have much higher mutation rates than those with DNA genomes.
Yeah, that's exactly right. And so in my lab, we're very interested in the evolution of these larger genome DNA viruses, like the pox viruses, as sort of our pet virus.
Mm-hmm.
And in that case, the mutation rates have been estimated to be a couple orders of magnitude lower. Although, interestingly, there are some recent estimates from, this is Eddie Holmes' work. He's now in Australia. He was at Penn State for many years. And they gathered some virus samples from the wild, from the field. So these were taken in Australia from the 1950s when this poxvirus called myxoma was released to control rabbit populations. This is a virus that it can be very deadly to the European rabbits that were introduced to Australia. And so there are these freezers then of virus samples from the 50s, and you can kind of sample then over the years that they were collected and then compare the virus genomes and try to infer back a mutation rate. And so as they looked from these what were in vivo samples collected from rabbits, they are estimating a mutation rate actually a little bit closer to some of the RNA viruses. And that remains sort of a controversy in the field, is how much lower are the mutation rates of these DNA viruses really? Or as this HIV paper points out, if they sample in vivo, so the samples they're looking at that we'll talk about a bit are from human patients infected with HIV, versus if you infect cell lines with the virus, you get a lower estimate of HIV's mutation rate. And so we might be seeing a similar thing in viruses sort of every stripe, where depending on where you sample, you'll see different mutation rates.
So traditionally, one of the main regulators has thought to be the viral polymerase, right? So which for DNA viruses is able to correct mutations or mistakes, but not for RNA viruses, right?
Yeah, that's correct. And so that's, when the traditional view for many of the RNA viruses is that they have these so-called sloppy RNA polymerases that's actually potentially an advantage for these viruses. As they generate more diversity quickly, then they can become these quasi-species that become very hard for an immune system to pin down and block, because it's sort of a moving target in terms of the evolutionary implications.
You can actually make polymerases of RNA viruses that are less able to make mistakes. You can make them more faithful, if you will, and those don't do well in competition experiments.
Yeah, it's really interesting, isn't it, to have that ability to do those experiments and see how evolution may have, in essence, tuned the mutation rates, or some of the ideas out there from studies like that.
I think even in bacteria, like similar experiments have been done, which show that mutation rates are selected for.
Yeah, absolutely. And then, another really cool step in addition to that is not only are mutation rates selected for, but also just the idea that diversity is selected for. So if you have these quasi-species or these viruses that have a certain set of variation or set of mutation in the population, that they are sort of adapted for facing the next challenge that they'll see as they move from host to host or continue to replicate. Some really interesting work in your neighborhood in poliovirus on recoding entire poliovirus genomes. So geneticists going in and making a lot of, in some cases, silent changes, that is, changes that don't alter the amino acid coding potential of the genes of the poliovirus, and seeing that those viruses are less able to adapt, and potentially a cool new source of live attenuated vaccines just by changing the coding or the variation within a population.
Yeah, we have had a ongoing discussion on TWiEVO about those papers, and there is a big controversy ongoing. I don't know if you're aware of it, but as to the mechanism of attenuation by those massive changes. These are changes that don't change the amino acid sequence, but just change the RNA. And there's some debate about whether it's codon bias or codon pair bias or dinucleotide bias. That's actually... Have you followed this or heard of this controversy?
I have, yeah. So it's really fascinating. And I think, and it illustrates a larger point of how we as virologists, as experimentalists, as evolutionary biologists, on things that you think might be settled, there are still these huge debates and these new patterns. So we think about, in this codon bias case, you think about what collection of 3 nucleotides are forming the codon, but these kind of emergent patterns of more complicated arrangements of now it's not just a single codon, but the two codons next to each other and how those 6 nucleotides arrange. Or I've seen other people thinking about 8 nucleotide sort of patterns, 10 nucleotides. And so there are all these sort of cool cryptic sources of variation that might influence how the evolution. Yeah, really cool. Yep.
All right, we better get into this paper.
Good point. We're running. Man, we could be running long. If we're
I hope people enjoy our chatting anyway.
If no one else does, you and I will.
Yeah, we do.
So absolutely. Let's get back to this paper. And so, as I said, they ended up sampling, I think, about a dozen or so patients. And what they were doing differently from some of the other studies was to actually sequence the DNA from blood cells where the virus, the HIV virus, had inserted its genome by reverse transcriptase activity. And they did this as a way to try to get a new measurement that didn't depend on collecting virus and sequencing RNA, where you might run into that sort of selecting on a phenotype problem. So the way
problem like making a virus, right?
Yeah, it's just a small hurdle, right? Because so what they're aiming for, right, is just how fast are these mutations accumulating, not the ones that are sort of selected on to keep a virus going. And so that's what motivated them to look in the genomes of the cells that were infected, the PMBCs or peripheral blood monocyte cells.
So these are patients with confirmed AIDS, right? And they can get a little blood from them, and then they can get DNA from blood cells, right? That's how it works. So basically, you just need to take a blood sample, right?
Yeah, exactly. And so they did that. They do the sequencing, and then they sort of take another trick here, which is they don't just count up every single mutation. Instead, they count up what are called nonsense mutations or stop mutations. So these are mutations that within the coding regions of the genes of HIV, the 12 or so proteins, that will code for a change in the codon. So instead of just coding for amino acid, it actually codes for a stop. And these are thought to be lethal mutations, which basically would disable the virus from being productive. And their motivation for doing that is that they want to they're not counting every mutation, they're counting a subset, but they're counting a subset that they think will not be selected on. So again, they're trying to avoid the phenotype. This thing will not be inherited because it can't be passed along. The virus is sort of a dead end for the virus. And then, so if you buy that that's a good way to do it, then from there, they'll estimate out of the mutations they count, how many possible mutations they could see. So now they're using this to infer a point mutation rate in a new way that sort of their hope is to sidestep some of those selective pressures or the phenotypic considerations to get an unbiased calculation.
Do you buy it? Are you okay with it?
I think it's an interesting idea. So you have, in any case where you're counting, trying to come up with unbiased mutation rates, there are going to be some trade-offs. So in a lot of organisms, not viruses, but slower evolving organisms, like let's say fruit flies, for example, when people have done these kind of experiments, or maybe it's easier to think about budding yeast. So when people think about doing these experiments, they'll try to avoid phenotypic or selection by doing their experiments. So they're basically growing a bunch of cells and then bottlenecking them down to a very small population and repeating that process. And the reason they're doing that is to bottleneck or to use small population sizes where you're just sampling more random mutations as opposed to letting natural selection work and sort of shape the composition of the mutations that are accumulating. So that's a bit of a trade-off because you're just the way you're doing the experiment. Viruses provide another challenge, which is their populations are immediately so large that you can't, it's more challenging to bottleneck them. And so I think the authors came up with a clever way of just counting the lethal mutations, these nonsense or stop mutations, as a way of getting around that problem. So I think that was a cool idea. So, and then what they find when they make that calculation, is that the number they come up with, it was pretty shockingly higher than compared to previous experiments where either the virus was passaged in cell lines or infected in cell lines, or people took sort of these other ways of counting out the mutations, collecting it from plasma instead of the cells and sequencing the RNA, where again, selection might be at play. And so I believe that old estimates for the mutation rate of HIV was something like 1 mutation in 10,000 base pairs, whereas what they were seeing was 1 in every 250 base pairs. So
1 in 10,000 would mean every genome has at least 1 mutation, right?
That's right.
Because the genome is about 11, 12 kb, I think, right?
Yeah, exactly.
And then this means 1 in 250 means many, many more.
Yeah, it's pretty mind-boggling.
Wow.
And getting back to that point of, evolution moves pretty fast.
It does,
depending on where you look.
So these nonsense mutations are assumed to be lethal, right?
Correct. Yeah, and the reason for that, just to be kind of explicit, is that if one of the genes encoding a protein all of a sudden stops before the polymerase can run through, or you can translate all the way through, you'll end up with a truncated protein that is non-functional, and that virus will be sort of dead on arrival, so to speak.
Okay. So do they use the same approach to look at the viral DNA?
Yeah, that's exactly right. So they're in terms of counting the
stop codons, right?
Correct, exactly. Yep. And they are, HIV it's mostly just coding potential, and so there aren't many intragenic regions, but that's the way they did it.
Mm-hmm.
So then from there, there's sort of a twist on the paper, because it turns out that you might think, based on sort of the estimates they're making, and we already hinted at this idea that the polymerases in a lot of these RNA viruses can be pretty mutagenic or sloppy, that's sort of the story, that the virus is just really churning out a lot of point mutations to perhaps diversify very quickly and avoid the immune system.
I'm sorry, I forgot. I misinterpreted. The 1 in 250 is for the DNA, right?
Oh, correct. Yeah, sorry.
I thought that was plasma. What's the plasma number?
Yeah. So the plasma number, in this case, they're collecting RNA.
Right.
And sequencing that, which is sort of how this had been done in past studies. And in those cases, their numbers match the older studies of a lower mutation rate, but they don't attribute that to the difference in sequencing RNA and DNA. They're attributing that to the fact that they're collecting the RNA from plasma as opposed from the cells. So in that case, what they're saying is that the virus, in order to get packaged, and this is actually, I'm glad you're bringing this up, Vincent, because it's a little bit hazy, I would say. They're trying to make the point that a highly mutated copy of the HIV virus won't get packaged into a productive particle,
right,
and make it out into the plasma. But I'm kind of curious about, does that imply a kind of a quality control mechanism, or what's going on there?
I don't know. I think some of the mutated genomes could be packaged. It depends where the defect is, right? I mean, the defect could be on an infection.
Yeah, exactly.
So if you're just sampling all the virus particles in the blood, it may not give you an estimation of what is actually infectious or not, right?
Yeah, exactly.
Or I should say it the other way, the lethal mutations, because you may have some in there that are lethal, but at a later step in infection.
Yeah, good point. So, and that would be perhaps interesting to go back into that data set and look, are there lethal mutations, that they see? But
the point is that in blood, if you take viral RNA, you get about 1 mutation per 10,000 bases, and in the DNA, it's 1 in 250.
Yeah, crazy.
And that's brand new. No one had ever known that before, right?
Yeah, that, kind of revising up 100-fold is pretty wild. And I think why this paper got my attention as well. It turns out, as I was starting to get to, there's a twist to the story, which is this is not just the virus polymerase, which is that those estimates are probably pretty clear, but what they do by looking more closely, or what they get to by looking more closely at the sequences and how they're mutated, turns out to be very revealing. And that's because there's one of the antiviral activities that humans and other mammals have are this family of RNA editing enzymes called deanimases, the so-called ApoBec family. And this is a defense, an innate immune defense, where basically these enzymes come in and see the single strand of DNA and start editing out base pairs to introduce hypermutations or more and more mutations. And in fact, the authors, as they look at, and there are clear patterns of how those mutations accumulate, and when the authors take that into account, they make an estimate that 98% of the mutations that they're measuring might be related to that ApoBec activity as opposed to the polymerase activity.
Yeah, it's remarkable. It really is.
Yeah.
So these
really
antiviral ApoBecs normally, they can be packaged in the particle, in which case they would cause extensive mutation of the viral DNA, right, and basically extinct it.
Correct.
But the virus has a protein that makes that get degraded. Otherwise, the virus would never go anywhere, right?
Exactly. And so this protein called VIF in HIV is acts to block the activity of ApoBecs. And
so what we are seeing is the effect of ApoBecs in the cell, not in the virus particle, right? Because they're looking at cellular DNA.
That's exactly right.
Okay.
Yep. And so it just shows you how in the cell, how active a process that is, and how important, basically, potentially, this process is for slowing down the virus. And in fact, they end up making... So they try to, at the end of the paper, they correlate this idea of differences in mutation rates, possibly, with differences in ApoBec activity in the patients. So the patients they're looking at, some are classified as normal progressors and others are classified as rapid progressors, meaning going from being infected with HIV to showing the symptoms of AIDS. And I would say it's a provocative idea that the mutation rate of HIV is sort of different in different people depending on how much ApoBec activity they have. But what they're trying to say, or what they're correlating or putting together, is this idea that in patients, the rapid progressors, that they see less of the footprints of mutational editing by ApoBecs, whereas in the normal progressors, it looks like that ApoBec activity might be a bit higher, and that this could be slowing down the virus.
They correlate that with a polymorphism in one of the ApoBec genes, right?
Yeah, exactly. And so this raises, I think, a really fun point about these host pathogen or host virus interactions in these so-called restriction factors like ApoBecs, is that, we've been talking a bunch about how diverse these virus populations are. But if we look in the hosts, including ourselves, at what are the genes that are evolving the most rapidly, ApoBecs stand out, and other restriction factors that are important for blocking retroviruses. And so this has spawned this, this field that I'm a part of, in this idea of kind of mixing experimental biology with evolutionary biology to think about the role of these genetic conflicts or evolutionary arms races almost, where you see this really dramatic rapid evolution between hosts and their pathogens.
Do we know what's been driving the ApoBec evolution in people?
Yeah, so that's a great question, and it's continues to be an active area of research. Folks like Michael Emmerman up at the Fred Hutch is continuing to work on this, and others. And people are starting to put some ideas out there in terms of what are the specific pressures in specific species. It's really difficult. So to really nail this down, so what we often do is sort of take a step back and consider the diversity not just of humans, but of all of our primate relatives when we think about these genetic conflicts. And what that does is oftentimes gives us a nice view of how rapid evolution is playing out, sort of a comparative, given the long generation times and small populations of mammals and primates. But one of the trade-offs is that then you start to lose resolution of what are the exact pathogens that might have been driving this evolution. And so what's happening now is that we're able, especially with experiments and more powerful genomic techniques, to start to look at this more closely and see if we can actually not only chart sort of deep evolution and these recurring patterns of genetic conflicts, but even coevolution between sort of our contemporary foes like HIV or other retroviruses potentially.
Do you think ApoBecs have any effect on poxvirus mutation rates?
Vincent, I thought you might ask me that question, so I did a quick Google search before our podcast, and there is a paper out there published, I think, Journal of Virology a few years ago, claiming no connection between pox viruses and apobex. It's kind of a cool idea that might be going on because pox viruses hang out in the cytoplasm, which is where apobex localize as well. So they might be at the same place at the same time.
Sure.
But no one has been able to see those same sort of clear marks that are very, very evident in the RNA genomes. And so I think it's really cool because it sort of, as I was mentioning before with some of Eddie Holmes' work, looking at these potential higher mutation rates of pox viruses in that case in a rabbit population, looking at from real animals. Is there another force at work that we just don't know about that could be influencing the mutation rates there? And I think that remains an open question.
Yeah, for sure. And I also would suspect that it's early days in terms of apobex and other viruses because we just haven't looked at very many, right?
I agree. Yep. But there's at least what the evolutionary analysis is telling us is that there has been a lot of things happening there as evidenced by the rapid evolution. And so that gives us this sort of this beacon or kind of homing signal to say, hey, maybe we should pay attention to this because there's a lot of really interesting things happening.
Yeah. Well, so it would have to be a cytoplasmic virus and it would have to be a virus with a DNA intermediate, right? That's
correct. Yep, for the apobex to work on that. It's also interesting to consider the other roles of apobex in the cell. So they're not only antivirals, as we've been talking about a bit, but they have some actually. So it's a massive family of genes, many genes in a growing gene family, which again sort of speaks to this diversity of the apobex family, or another level of mutation that is sort of consistent with these genetic conflicts or arms races. But at the same time, there are these just housekeeping functions that different members of the apobex family provide. So maintaining nucleotide pools, so doing pyrimidine salvaging, which is an important just sort of housekeeping role of keeping the nucleotides that are used for DNA replication active, and then there's filled, and then there's also this protein AID, which is involved in the diversity of our antibodies. So there's this process of somatic hypermutation, where in order to generate the antibodies that are part of the adaptive immune system, you have to very quickly and without sort of the luxury of going through many generations, make a lot of point mutations in these these genes that encode the antibody chains. And so we've employed these apobex, or a gene like apobex basically in this family, to assist with that. And so it's really cool, right, to step back and just look at all of this sort of extracurricular mutational activity that we depend on.
Yeah. I think you should tell our listeners what apobex stands for, Nels.
Oh, another gotcha question. Let's see. It's something like apolipoprotein and then something. Pretty
apolipoprotein B, mRNA editing enzyme, catalytic polypeptide-like.
That sounds pretty clear. Yeah.
What a name! I like apobex.
It's a much more easy name to pronounce or to think about. Yeah.
So this idea that you mentioned that these apobex, which we consider antiviral, could also contribute to variation and therefore make, more serious disease in some patients versus others. This is really interesting.
Yeah, it's a very cool idea. And I think for me, that was one of the things that made this paper fun to read. At the same time, I want to also maybe put this forward as a potential shortcoming of the study, because now if we're defining a mutation rate of HIV, of a virus, based 98% on the activity of a host gene. And if that host gene is variable, so that you might have different activity depending on the host, how far have we come in really defining what a mutation rate is if it depends on all these variables? And so I think it raises kind of an interesting point that we were discussing earlier, which is, as evolutionary biologists, population geneticists, folks spend a lot of time worrying about getting rid of selection and thinking about, how do we measure just the accumulation of mutations as opposed to the influence of natural selection on them. And here, by reinstituting the ApoBec activity, which itself is very variable, and under all of this natural selection on the host side, we've sort of muddied the waters, I think, of what a mutation rate is in just sort of a strict definitional sense.
But remember, even if you took away the 98% mutations due to ApoBec, you still have 1 per 10,000 bases, right?
Yeah, absolutely, which is pretty wild to begin with.
Yeah, so that, I mean, we've always considered that to be very high, so this is just making it even more serious and bringing in the possibility of other viruses tapping into other host proteins. And I'm sure now going forward, people are going to be taking the different categories of AIDS patients, you know, the normal ones, the short-term ones, the long-term rapid progressors, and the elite controllers who live for years, and sequencing their ApoBec genes and now seeing what we can pick up, right?
Absolutely. And don't get me wrong, I think the paper is really fascinating. And it makes an important point by revealing the influence or the importance of this gene editing activity that pushes the mutation rate even higher.
Yep.
It's just kind of keeping all of these things in mind as we make comparisons.
I think it's a paradigm shift in a way, right?
Yeah, absolutely. Pretty provocative to think about how littered with mutations these viruses can get.
Yep. It's cool stuff.
Yeah.
Anything else that we need to mention before we move on?
Yeah, I think that about covers it for today's paper. But just to sort of maybe close on this idea again of how much time evolutionary thinkers spend worrying about mutation rates or, describing and getting a handle, characterizing these things. This is one of the major pursuits of the field, is to understand the sources of the variation that we see all around us. And so this is, I think, one of the main pillars of doing that. And so what we see with today's paper is just a very recent, published about a month ago, effort to do that, and it just highlights how this is going on not only in viruses, also in other microorganisms, and then with the increasing power of our genomics reach in bacteria, and then species with larger genomes like ourselves.
I mentioned earlier this paper about VSV plaque-to-plaque variation, and now I realize that would be a perfect paper for TWiEVO.
Yeah, I agree.
So take a look at it, see if you like it.
I will. Thanks.
You don't have to do that on TWiEVO because you're the guy who should be talking about it.
Well, you could always cross-pollinate. Maybe I could show up, make a quick appearance on TWiEVO from time to time as well.
You're always welcome to do that. And now that you have a great Skype set up there with your wonderful mic and headphones, yeah.
Yeah, I'm ready to go.
Good. Let's do some picks.
Great idea. So my pick of the week, science pick of the week, is actually a local pick, and it is more about natural history. So as I'm sitting here in the Elde Lab studios, I'm looking out the window at the Great Salt Lake in this really cool state park called Antelope Island. It's about a 45-minute drive from the University of Utah here.
You can see it from your window?
Yeah, I can. I can see the peak that I've hiked to from time to time.
Nice.
It's in the Great Salt Lake. Really fascinating park that has some native species, things like pronghorn antelope, all kinds of badgers and birds and wolves and coyotes and so forth. The other thing that Antelope Island has, which makes it pretty interesting, is an introduced population of bison that was, I think it was more than 100 years, 7 bison showed up, and now there's hundreds of them out there. It's really crazy to be going on a hike and run into a 1.5 ton bison on your hike. The reason I'm using this as a pick this week is just this last weekend, the bison were rounded up in an annual tradition where the bison are brought together. They cull the herd. They actually auction off a couple hundred of these things. So people, restaurants will do this, farmers will do this in order to get bison meat. And I've actually been threatening some of my colleagues of trying to get a group together of about 10 or a dozen of us to try to buy a bison at the auction and then split up the meat after it's butchered. If you're interested in just kind of seeing the nuts and bolts of this, it's turned into sort of a tourist activity. So the New York Times actually covered this last weekend's bison roundup. And you can see some of the fun of being in the Salt Lake region and kind of getting back to the western frontier, at least for a weekend or two.
It's pretty cool.
Yeah, from an evolutionary, it's kind of a stretch, but from an evolutionary standpoint, what we're seeing is a big artificial selection experiment where, as the veterinarians take the rounded up bison, they're making decisions about which in the population to remove and which to release back in order to try to keep a healthy, balanced herd over the years. And so sort of this interesting idea of artificial selection. Who are we selecting for versus what might happen naturally if the bison were just allowed to run over the island?
I don't know. In my experience, when humans apply selection, it's always bad.
I think you can mount some pretty good evidence for your argument there.
They also vaccinate these guys. The ones who aren't taken for meat, they vaccinate?
Yeah, that's right. So again, trying to manage a healthy herd. And so they take all kinds of measurements. They're looking for signs of disease or parasites. And actually, I'm not sure the specifics on the vaccines. That would be interesting to know the virology angle.
They don't tell you. Could be bacteria and viruses, right? I don't know a lot about bison viruses, that's for sure.
I don't either, but I'm tempted now to head out this weekend. They actually let you sort of kick the tires of the bison, so to speak, a week before the auction. Maybe I could get in the ear of a veterinarian and find out.
It could be they're just selecting for the faster bison that can't be captured.
That's right.
or not dumb enough to go whatever they're using to attract them.
That's right. It's that wily bison that you want as your as sort of the star of the herd going forward.
It's they say they're very fast here.
They really are. And I have to tell you, Vincent, so I've been on a few hikes where you come, first of all, Antelope Island is sort of otherworldly. If you go to we'll put up the park website as well, and there are a couple snapshots from it. And so you're surrounded by the Great Salt Lake and then mountains kind of on all sides. But at the same time, it feels like you're walking through a prairie. And all of a sudden, you'll come up and maybe 20 yards in front of you is a massive male bull bison just standing, staring at you. And you will stop in your tracks and sort of let the bison decide what it wants to do before you go on with your hike. It's really fun.
So I take it you read the New York Times, eh?
Yeah, from time to time.
Yeah, online, right?
Oh, yeah. Yep.
Is there any local paper that you read?
Sure. So there's the Salt Lake Tribune is the main one. It's the more liberal of the two regional Salt Lake papers. There's actually a great political cartoonist, a guy named Pat Bagley, who a lot of his cartoons will show up in newspapers all over the country. I'll probably make that a pick down the road a little bit, some of his really interesting and progressive cutting cartoons. So again, Salt Lake, I think, is known, mostly for some of the religious and conservative aspects, but there's really this great progressive undercurrent here. And then, of course, in addition to being in a increasingly interesting city, we're surrounded by all of these beautiful areas, such as Antelope Island.
Yeah, it's pretty nice out there. And if I remember, your university is kind of up on a hill, right?
Yeah, exactly. So we're just sitting here in the foothills of the Wasatch Front.
And that's why you have a good view of that lake, right?
Yeah, exactly. And with within range of ski resorts and mountain bike trails and everything else. So we'll get you out here soon, Vincent, for a visit.
Love to, yeah. It'd be fun.
Yeah. How about you? What's your pick of the week?
Well, my pick has to do with carcinogenesis that you mentioned at the very beginning of this show. The idea that, you have to do something to fruit flies to get mutants. And people are very good at mutagenizing ourselves. A lot of the things we surround ourselves with and eat turn out to cause cancer. They cause mutations. And, the idea is you need maybe 10 or 15 mutations in a variety of different genes, and then your cells are growing uncontrollably, and then they become a tumor. Things like, I guess, asbestos would be one that comes to mind, right?
Yeah.
The chimney sweeps from the 1800s who used the little kids, they would get to fit down the chimney. They would wear shorts. And they got testicular cancer because of the tar on the wall of the chimneys. All this stuff. It goes on and on and on. Well, here's an article in Ars Technica. Bacon, hot dogs, and other processed meat cause cancer. WHO announces this. It's based on a paper published in Lancet Oncology, and it's linked to in the article if you want. But it's basically a meta-analysis of 800 different epidemiological studies. So these are observational, where they look at what people are eating and what kind of cancers they get. And, there are things in bacon and processed meats, various chemicals which are probably implicated in this.
Yeah.
The study found that every additional 100 grams of red meat eaten daily increased the relative risks of colorectal cancer by 17%. And other things, meats that are made into cold cuts and they're smoked, they're cured, they're carcinogens like N-nitroso compounds, polycyclic aromatic hydrocarbons. And, when you grill meat, it transforms some of these chemicals into worse carcinogens, so.
Yeah, which also correlates with how delicious the meat is, right?
Yeah, I'm looking at this picture of bacon here, and I don't eat bacon very often because it is a high-fat containing meat, but when I do it, it's just delicious, and it's too bad now I won't eat it at all, I suppose. Well, I suppose if you just eat it once a year, you'll be okay.
Well, yeah, it's an interesting point, Vincent. I think you're trying to set me up here for something, but I've eaten bacon a couple times since I already read this article.
Yeah.
And processed meats. And part of it is because, well, it's super delicious, and how do you want to live your life eating twigs and sort of living long but kind of withering away. Or the other thing is how this is a great illustrative point, I think, about how do we balance, what do these risks really mean, and how do we balance that against what risks are just out there anyway. And so you live in New York, and I can imagine as you walk home this evening, there's some negligible risk that you could get hit by a car.
Oh, sure.
I live in Salt Lake City, and when I go hike on Antelope Island, there's some negligible risk that a bison will charge me down and take me out.
Sure.
And so how do we weigh that negligible risk, which we'd probably take without even batting an eyelash, to this risk of eating a little extra bacon or a hot dog here and there in the whole scheme of things? And so I would caution a little bit of, it's definitely a provocative story. It's kind of blowing up on my Facebook page and on social media. But I think we're already seeing some interesting counterpoints about how do we actually take the numbers that are out there that can sound pretty scary and actually kind of make sense of them.
Yeah, I think driving is really a dangerous occupation, right? People are dying all the time.
Oh, man.
In driving accidents. So that's much worse than eating bacon. I would say that, if you eat it every day, you probably ought to cut back. But probably once a week is fine. Oh, yeah.
I think so, too. And, I think there's a line, what's that? There's a movie actually about the West with Billy Crystal, and he says, that guy ate bacon every day. And I think after he died or something like that. But anything in moderation. The other point I would make on this is that the World Health Organization, I just saw an article, kind of one of these counterpoints, and they have these categories where they have taken now, I think, processed meats and put it in the same category as smoking, as in terms of cancer. Mm-hmm. And the point this article made, which is it's a subtle one, but an important one, is that rating does not actually reflect the actual risk of smoking versus eating processed meats. What it reflects is the certainty of the evidence. And so the processed meats could, raise your chance by some really small amount, whereas smoking, actually, I think there is very good evidence that long-term smoking is very strongly associated with risks for lung cancer.
Right.
But the fact that they're shared in that same category, I think, also raised a lot of eyebrows, where in fact it's more about the evidence than sort of the actual impact or influence of these things. Right. Yeah, it's an important distinction.
So many years ago, both my parents died of colon cancer, right? So I decided not to eat red meat. My wife and I. So her father actually also died of colon cancer. So we decided to cut out red meat. So for about 5 years, we were pretty good. And then we said, it's just too good.
Life's too short, as they say.
So we do moderate. Maybe once a month or something. But, it's all a matter of risk and how much you want to worry about it, and how much you, as you said, how much you want to enjoy your life. And I've already passed my father in age, so I figure I'm doing all right.
Absolutely. Of course, there are these genetic components. Again, we're still our infancy of understanding how that all contributes. And so the jury, I would say, is still out there. And there are other actually great reasons for moderating or cutting back on red meat that have to do with environmental implications, right? How many resources does it take for us to farm and do these things? So, yeah.
Also, what was the other... I've lost it, but maybe it'll come back. But in the meantime, of course, it's good to drink red wine, and so I've embraced that very much.
Good idea.
Oh, yes, so you can get screening for colon cancer, right? So you can pick it up early if you have a screen every 4 to 5 years. So I do that. Everyone past the age of 50 should do that.
Yeah,
and the screening will get better and better. We can't, unfortunately, pick up every cancer that you can get. I talked with Blossom Domania a couple of weeks ago on TWiEVO. We were talking about Kaposi's sarcoma, which can happen not only on your skin, but inside of you, and you can't pick it up until it's too late.
Yeah.
But, people are working on trying to make better diagnostics, and someday we'll have them.
Yeah, another active area of research. How often and when should we be tested? In some cases, some counterintuitive findings, but we're getting better at figuring out how to move forward.
As I've said before many times, someday we're going to wake up, stand in front of your mirror, which is actually a very sophisticated sensor. It's going to do a detailed scan of your whole body and then say, have a nice day, you're fine, or you should go see the doctor. I've already sent him or her your image. I see a little spot here. It's going to happen.
Yeah, it sounds like Apple Mirror.
Yes, Apple Mirror. It's going to be very slick looking.
There we go.
Now, if you would like to contribute a listener pick, you should. We love getting them, and you can send them to TWiEVO, TWiEVO at microbe.tv. Hopefully by the time we launch, that website will be active. Well, actually, the email will be anyway.
Yeah.
And if you have questions or comments, you should also send them to us, TWiEVO at microbe.tv. You'll be able to find TWiEVO at iTunes and at microbe.tv/TWiEVO. If you're wondering, microbe.tv is my new home for all my podcasts. I hope to accumulate them, and then also to get other people's podcasts there as well. The ones that I think are a good fit, we'll get them and make them part of the Microbe TV world. And I want to thank you, Nels, for doing this. I really enjoyed talking with you. I learned a lot.
Thank you, Vincent. This is really fun. I can't wait for the next time.
You can find Nels at cellvolution.org. He's also on Twitter, ELE Early Bird, so that's like EL with Early Bird after it, right?
Correct.
How would you pronounce that?
L Early Bird.
L Early Bird, one word, that's right. Check it out. And I'm Vincent Racaniello. You can find me at virology.ws. I'm also on Twitter. I am Prof. VRR. You can't say it, but you get the idea, right?
Yeah.
And the music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. I bet it wasn't hard to get that domain name, right?
Yeah, pretty unique.
Been listening to This Week in Evolution, the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.